Hey guys, welcome to question 2 of the Unit 1 FRQ progress checks. Um, this one also corresponds to the chemical basis of life. So again, chapters 3, 4, and 5, um, water, carbon, macromolecules. Um, so let's take a look at question number 2. So scientists are studying the structure and function of receptor X, a single polypeptide protein that is found in the membrane around certain types of cells. Receptor X contains no alpha helices or beta sheets. A specific molecule outside the cells is recognized and bound by receptor X. The binding of the molecule to receptor X causes the cells to have a particular response. To study the structure and function of receptor X, the scientists are altering the sequence of small sections of the receptor. Both the normal sequence of a 5 amino acid section of receptor X and an altered sequence that contains 2 amino acid substitutions are shown in figure 1. So there's a lot of different stuff that's being assessed here. So first of all, you've got cell signaling, which is the context in which they are assessing your understanding of amino acid structure and stuff like that. So let's take a look at what the actual questions are asking. Okay, identify the process used to form the covalent peptide bonds that join amino acids into a polypeptide. Okay, well, the important thing here is that you know that all macromolecules, in this case polypeptides, are going to be, formed from monomers. Right, so monomers, so in this case, the monomers are amino acids, and they get formed into polymers, which in this case is a polypeptide. And okay, and the way that happens is going to be through this process of dehydration synthesis. And what dehydration synthesis is you basically take two monomers. So if this is an amino acid, right, and this is one end of the amino acid, what you do is you're going to take an H from one and an OH from the other, and then an H2O is going to be released every time one of those is made. It was one of the very first things we talked about when we started our Unit 2, biochemistry and cell biology. And you guys have modeled that before in class, although it was a while ago. Okay, so that's really about it for question 1. And it's the same no matter what it is. Amino acids into polypeptides, sugars into monosaccharides into polysaccharides, nucleotides into nucleic acids. Anytime you get monomers into a polymer, it's always going to be dehydration synthesis. Reverse is going to be a hydrolysis, where you take H2O, you put it in, one hydrogen would end up here, one OH would end up here attached to the sort of carboxyl end of the amino acid. And one of those is going to happen sort of at every polypeptide. Okay, at every amino acid in the polypeptide. Okay, let's take a look at B. The change in the amino acid sequence is caused a change in the shape of receptor X based on the R groups of the original and substituted amino acid. Explain why receptor X changed shape. Okay, well, so if these are the amino acids, and it looks like these are the amino acids that were actually changed, because it looks like this is the same. What you really have to remember here is that first of all, you got to know what an R group is. And an R group is as we talked about last semester, is this set of this area over here that is different, right? Because other than the R groups, they're all the same, right? They've got their amino N, their carboxyl N, that hydrogen, and this is the only thing that actually makes the amino acids different from one another. It's what makes alanine, alanine, and threonine, threonine, and tryptophan, tryptophan, and all. Basically, every amino acid is going to be unique because of these R groups. And so when you change the R groups, you're going to change the structure of the protein because they're going to interact differently. These, you can see, are charged, or ionic, right, or polar, right, however you want to say it. But you can tell that they've at least got partial charges, if not full charges. So they're going to be, for example, hydrophilic. And they're going to just interact differently. Whereas these have now been replaced with nonpolar ones, right? No charges, no anything, just, 3 hydrogens coming off, right? So nonpolar, those are going to be hydrophobic, right? And they're going to want to interact with nonpolar stuff. So depending on where these R groups want to go, that determines the three-dimensional shape of the protein, which is the tertiary structure, the shape of the protein. So R group interactions actually lead to protein shape. So so different R groups are going to lead to a differently shaped protein. And again, so if we just look at the scoring guidelines, dehydration synthesis, right, or condensation reaction, which is another name for the same thing, 2 amino acids with charged or ionic or hydrophilic R groups were replaced with 2 amino acids with uncharged or nonpolar or hydrophobic. So I guess they're not polar, right? So charged or ionic, hydrophilic, either way. I guess they have full charges, not partial, but so charged or ionic. Okay, and then, nonpolar, hydrophobic. So now we're all set up to answer question part C of question 2. And let's say using the template below that represents the 4 levels of protein structure, place an X on each level that is expected to be altered as a result of the amino acid substitutions shown in figure 1. Okay, well, so we can clearly see that the primary structure, which is just the order of amino acids, that's clearly changed, right? This one's different from this one, and same thing, this one's different from that one. So clearly the primary structure has been changed. And then from our answer, because these R groups have been changed, right? These R groups are right here. They've been changed, so that is definitely going to change the tertiary structure. So we're going to put that there. Now, I'm going to only put X's there, and I know some of you are saying, well, wait a minute. I remember taking the macromolecule quiz, and there was that question that said primary structure, a change in primary structure is going to change secondary, tertiary, and quaternary. A change in primary is going to change all the others. That is true, and especially in the context of the quiz question where you answered that, that was extremely true. But there's a couple of things in here that are actually you need to pay attention to. So one of the things it says is that receptor X is a single polypeptide protein. Okay, so when it says that, what that means is there is no quaternary structure, because quaternary structure is more than one polypeptide coming together to make a protein. It's common. Lots of, like, hemoglobin has 4 polypeptides. But it's some proteins do not have quaternary structure. So that's what tells you that there's no quaternary structure. And then also it says that it contains no alpha helices or beta sheets. And so since it contains none of those things, then it contains no secondary structure, because that's what secondary structure is. So that's why I didn't put X's there and there. Explain how the amino acid substitution is most likely to affect the function of receptor X. Well, we've talked a lot about how structure determines function. So if you change the shape of the protein, you're going to change the function of the receptor, because receptors are specific for a molecule of a particular shape. Specific. Okay, to a particular shape. And if you change the shape of the receptor, you're going to, then that receptor is no longer going to be receptive to a particularly shaped molecule. Okay. All right, so again, yep, primary structure, tertiary structure, and then function is determined by shape. The function of receptor X is likely to change, or the structural change might alter the binding of a molecule to receptor X. We kind of addressed both of those. Structural change might alter the ability of receptor X to cause a cellular response. Sure, same kind of deal. Cool. So that is question 2. One other thing that I want to kind of clue you guys into is, if you're thinking about time, or like not writing too much, one thing that you could do is I kind of modeled it a little bit, is you can sort of plan out your answer and then put these in complete sentences, which, because of time, I'm not going to like type out the full sentences, but you can see how you could take this information and easily get it into a couple of sentences and then move on, rather than just sort of starting to write and then, letting the answer take you where it wants to go, which some of you do sometimes. It might be easier to plan out your stuff in advance and then put it into to sentence form, especially if you find yourself struggling with time or writing more than you need to. Okay, so we'll come back for Unit 2, where we'll get into cell biology. Okay, thanks.